down here.
Into the matrix.
So it goes through the two membranes of the mitochondria. We have the outer membrane, the inner membrane space, the inner membrane, and the matrix. Pyruvate goes through both. It has to go through what's to get there?
Little
Transport proteins. Okay, it just goes through transport proteins. What mechanism gets it in there? Is it active or passive?
Passive.
Is it simple or facilitative?
Facilitated.
Inside, we have pyruvate. Pyruvate gets converted into an acetyl group, which is 2 carbons from 3 carbons, by the removal of
CO2.
CO2. Every time we have CO2, what do we make? An ADH. Now we've got an acetyl group, which is a 2-carbon group. Right? 2-carbon group needs to join into the Krebs cycle. Right? Problem is the acetyl group can't get into the Krebs cycle on its own. It needs a helper. What's the helper called? Coenzyme A. Okay. The Krebs cycle is starting off with a 4-carbon material called OAA, which stands for one go for 2.
Oxaloacetic acid.
Oxaloacetic acid. All right. OAA. We're going to add our acetyl group with the help of coenzyme A, which is going to come on, make acetyl-CoA, and then coenzyme A is going to leave after it attaches the acetyl group to the OAA. Okay. When you attach acetyl to the 2-carbon acetyl to the 4-carbon OAA, right? What's the first stable compound?
Citric acid.
Citric acid, which is what the cycle is named for. Or Krebs, from Hans Krebs. Citric acid, how many carbons?
6.
This is 4. This is 6. This is a cycle, so we got to get back around here to OAA. In the process of going from 6 carbons back to 4 carbons, what do we need to release? 2 carbons. 2 carbons come off as CO2. When they come off as CO2, what else comes off?
NADH.
NADH. Okay, at this point we'd be back down to a 4-carbon material, but that 4-carbon material is the wrong shape, so we got to rearrange it to get it back to OAA, right? What do we need to do to rearrange it to get it back to OAA? What comes off?
An ATP.
We're going to make ATP. We're going to make FADH2 and another NADH. How many times do we run the Krebs cycle?
Twice.
Times 2. So for every run of the Krebs cycle, we're going to get how many NADHs? 3 plus 1, 2, right? 4, 5 times 2 is 10 NADHs. How many ATP at this point? 1, 2 plus 3, 4, 5, 6 minus the 2 that started the whole process. So at this point we've netted 4 ATP, right? And we've got 10 NADHs out. Great. Now what do we do? Right? The NADHs come over to the electron transport chain. So here's our membrane. Right? We've got the electron transport chain. What? You don't like my drawing? Right, so this is the outer membrane, this is the inner membrane space, this is the inner membrane, and the matrix. In the matrix, what do we make over there?
Lots.
NADH. Okay, so NADH is going to come over. It's going to go back to NAD plus, right, by dropping off what? 2 electrons and
Hydrogen.
a proton, right? The electrons are going to power the pumps. What are the pumps going to do?
Pump protons in the inner membrane space.
Pump protons into the inner membrane space, building up a what?
High concentration in the inner membrane space.
High concentration of protons in the inner membrane space, right? This will be a low concentration in the matrix. Which way would the protons like to go if they possibly
Back
could?
in the matrix.
Into the matrix. So the protons will go into the matrix, and in the process we will make
ATP.
ATP. How is it made? SLP or OP?
OP.
OP.
Oxidative.
Oxidative phosphorylation. But exactly. Okay. Now, in order to keep this whole thing running, it is dependent upon the presence of what? Oxygen. So 1/2 of an O2 is going to come into this last one, and it's going to leave as H2O. So what's the difference in an O and an H2O? What does it need to pick up?
2H.
It needs to pick up 2 H pluses, and it needs to pick up the electrons, and it leaves as water, and that keeps the whole thing flowing. Yes, sir.
So why does it, if it's just picking up the 2 H's, why does it brush by the last of the transport chain?
Just getting them to
It's picking the electrons off the last of the last electron carrier. Okay, it's like the runner on third going home, so that the runner on second can go to third, the runner on first can go to second, and the guy who's batting has somewhere to go. You gotta keep them moving.
So oxygen is the last acceptor.
Oxygen is the final electron acceptor.
And then over there, through the membrane,
Yep.
it's passive transport.
Passive transport facilitated diffusion through some carrier proteins.
Over here you pick up 2 hydrogens,
Yep.
but in photosynthesis, don't you only let go of 1 hydrogen?
You're dropping off, if oxygen's leaving, you gotta be dropping off 2 of them. I probably just drew it with 1. So you're dropping off protons, you're dropping off electrons. Okay? Now, all of this works great as long as what is present?
Oxygen and water.
Oxygen, right? As long as oxygen is present, all this works great. What happens if oxygen is gone? If we got no oxygen, right? If there is no oxygen, is there anything to remove the hydrogen ions here? No. So they will continue to build up, and what's that going to do to your gradient?
Destroy.
It will diminish it, and it will not want to move anymore. Also, if there are no oxygens here, is there anything to remove the electrons?
No.
No. So is there anywhere for the electrons that are on NADH to go? No. So NADH has nowhere to drop off its electrons. If all of the NAD pluses pick up electrons and don't drop them off, pretty soon all of the NAD pluses are going to be in the form of NADH, right? And you will have no available NAD pluses to come in and remove them from the Krebs cycle. And then you'll back up, and there'll be no NAD pluses to come in and remove electrons from the grooming step. And now you back all the way up outside of the mitochondria, back into the cytoplasm. And is there a backup pathway that can keep this thing running?
Fermentation.
Okay, there are 2 of them, right? One of them is going to make lactic acid, and it's going to take in NAD plus, and it's going to make NAD plus from NADH. That's the critical point. That's the important bit, right? Is it's recycling the NAD plus. In yeast, we make acetaldehyde, and then acetaldehyde gets converted to ethanol by releasing a CO2 and recycling the NAD+. So what do yeast make? CO2 and alcohol. Okay, CO2 makes the bread rise. Okay, carbonates the beer.
So then, like, if you put yeast in your bread, are you, like, basically giving your bread alcohol?
Except that you bake bread in an oxygen.
Or,
not oxygen.
okay.
Right.
Yeah.
So, we go over like the poisons and stuff.
Yeah, so there are a lot of poisons that can act on various things. Okay, they interact over here. One of them is called BNP. BNP is the one that makes the membrane leaky.
Do we need to know the difference between the poisons, or do we need to just?
Not really.